Thank you for the organizers for inviting me. This is a great honor. So I will talk about studying drug resistance, and I'm not talking specifically about AI, but I'm talking about many statistical methods, which is the basis of AI, and which comes from machine learning, which is actually a part of statistical modeling of data. So these are my disclosures. Get research funds from IBM, and which we've related to later. So what is cancer? We all start from a fertilized egg that potentially could have germline risk alleles. Cells divide. As they divide, they accumulate DNA damage. Most of the damage is repaired over time. But if it's not repaired, it becomes mutations in the DNA, and those occur anywhere in the DNA. Mostly they don't do anything. These we call passenger mutations. And occasionally there could be a mutation that increases the fitness of the cells, and the cells start to grow faster or die slower, and they accumulate. And that's a clonal growth. This is not yet cancer. It's a clonal growth. Then maybe the cancer accumulates more mutations, starts to grow, or the lesion or the growth accumulates more mutations, and starts to grow even further. At some point, the immune system might notice that lesion and fight it, and those lesions would never become cancers. Some evade the immune system and eventually grow and become cancer. And then cancer is diagnosed, and when it's diagnosed, it already has thousands of mutations throughout the genome. It could be also hundreds of thousands, depending on the tumor type, and also point mutations, single base changes, or chromosomal changes or rearrangements. And that's when we diagnose and typically analyze the cancer genome. Then hopefully we treat the patient, and hopefully most of the cells die. But maybe there was a subset of cells that had a mutation that confirms resistance to the therapy, and then that clone continues to grow and becomes a recurrence of cancer. And then another treatment maybe is given. And in this case, in this scenario here, even during treatment, a new mutation arises that give rise to resistance to the therapy, and now another recurrence occurs, and that's kind of the life history of cancer. And here's kind of a very known picture to show this kind of history. This is a metastatic patient published by Nick Wagley several years ago that had melanoma with BRAF mutations, and then was treated with verapamil. And after a few weeks, you see there's no sign of the cancer, and that's really amazing work of the verapamil. But then a few weeks later, the cancer came back. And that's basically essentially the problem we're trying to hit, is how do we delay or prevent resistance? So how can we think about doing that? One way to do that is to take a lesson from HIV treatment with giving combination therapy of multiple drugs, with the idea that the drugs will have non-overlapping resistance mechanisms. Because if they have overlapping resistance mechanisms, do I have a pointer or something? I don't know if that's, I don't have a laser pointer. But if on the left, you can see a picture showing the world of all possible changes in the cells, and there are three circles. Each circle represents the resistant states of the cells to each particular drug. Now, if those circles overlap, then there's some states of the cells that they are actually resistant to all drugs, and therefore they will grow and become resistant. But if you pick three drugs, for example, in this picture, that have non-overlapping resistance mechanisms, then the cells have no way out, because whatever state they become, or whatever mutation they have, there's no way they could become resistant to all the drugs. And therefore, hopefully resistance will be delayed or even prevented. And so in order to go about doing that, we have first to map for every drug what are the resistance mechanisms for each drug, so we could actually draw these kind of imaginary circles and see if they have overlaps or don't have overlaps. So to do that, we need to build these maps like this one. And this is the most accurate map that exists today for anti-EGFR treatment in lung cancer and gefitinib. And what you see is that the majority of cases, when you have this, you have mutation in the EGFR target itself, the T790M, And in some cases, you have other events, like MET amplification, or HER2 amplification, or PIK3CA mutations, or BRAF mutations. But in other cases, you have transition to other cell states, like EMT, epithelial mesenchymal transition, or even going from non-small cell to small cell. And there are 18% of cases which we actually don't know what's the resistance mechanism. And this is the best map we have today. Most drugs and most tumor types, we have no clue what are the resistance mechanisms. So how do we find resistance mechanisms? So luckily, we've been working on finding drivers for more than a decade now, of primary tumors. So TCGA and ICGC, the TCGA is the national, the largest national project for studying drivers or cancer in general, and ICGC is the International Cancer Genome Consortium, which basically has similar projects around the world. They're all focused on primary tumors. What we need to focus now is on the end of that life's history of a cancer and understand what are the drivers of resistance. Now, as I said earlier, there are many, many mutations when we identify a cancer, but only maybe a handful of them are the drivers. Same goes for resistance. There could be additional mutations and variation that caused in the cells, but only a small fraction of them, maybe one, is actually responsible to the resistant. And it doesn't have to be a DNA mutation. It could be epigenetic change or cell state change. So over the years, we developed many tools, statistical tools, to analyze genetic data to understand what are the drivers. And the basis of all these tools is to generate a statistical model of what events we expect by chance, the passengers along the genome, and then look for genes or regions of the genomes that have more mutations than expected by chance, or a pattern of mutation that is unexpected. For example, clustering of the mutations in a particular amino acid or a region of the genome or a part of the protein. And we developed these different tools to identify those drivers. Out of the 1,000, could be millions of mutations across many patients, we want to identify the few genes that have those mutations that are actually selected for, and all the rest are not selected for. So we could apply the same notion for finding drivers, and go through three different ideas of how to study drivers. The first one is kind of the most simple approach to think. You take a sample before treatment and then after resistance, and you look for all the events that changed between the before treatment and after resistance, and you make this kind of what we call the commute plot. And then you ask, well, are there genes that have more mutations than you'd expect by chance, where now the background model is not the passenger mutations that accumulated from the fertilized egg, but the passenger mutations that accumulated from pre-treatment to post-resistance. So that's actually good for us because it's a lower background rate, and therefore it gives us more statistical power to find those drivers. But still, we need many samples to become powered to find those different resistance mechanisms, and that's why we need you to get those samples. So we could apply the same tools on these delta mutations. The next idea, which is a bit more complicated but more powerful, is to look at the dynamics of clones. So not only take 2 time points, but take multiple time points along the development of the cancer, and then look at which clones within the cancer are growing, and then look only at the mutations in those clones to see which are the drivers. So now I have kind of a open parenthesis here to tell you about a bit of the history of how do we analyze dynamics of cancer. So when you take even a simple single cancer sample, it has normal cells and tumor cells. And the first thing we need to know is what fraction of them are tumor cells out of all the cells. And, thank you. And here's a cartoon of a tumor sample that has 2/3 cancer cells, 1/3 normal cells. And there's a mutation there that exists in half of the cells. This mutation also occurs in a region that has 4 copies in the tumor, but of course there are 2 copies of the DNA in the normal in this chromosome. So overall, there are 10 copies of chromosomes, and 1 of them is mutated. So if we were to sequence this tumor out and to a depth of 100x, only 10 of the reads would have the mutation, and 90 reads would not have the mutations because they would be from all these other alleles that are not mutated. So that's on average, of course. So now we invert this logic. We sequence tumors, we find the allele fractions of different mutations and different copy number events, and then we calculate what is the cancer cell fraction of every mutation, meaning what fraction of cancer cells have that mutation. And as you can see, some mutations are clonal, meaning they're in all the cancer cells, but some mutations are subclonal. And now the fact that you see these distributions is because the data we have is limited, is basically the number of reads is a discrete number. So there's an uncertainty on the actual cancer cell fraction. Now, luckily, cancer, the mutations come in packs. Basically, all the cells that belong to a clone, all the mutations that are common to those cells should have the same CCF. So we could apply a Bayesian clustering algorithm to identify what are the clones that exist in this sample. And from going before the Bayesian algorithm, which we call Phylogenetic, after we find this. Again, many mutations are clonal. They're on the side that I can't use the laser to point to. But here, there are some subclones that are in subsets of the cells. There are mutations that are in 70% of the cells. There are some mutations that are in 45% of the cells, in 30% of the cells, etc. Now, even with this single sample, we could start to build the phylogenetic tree of that cancer, because those cells that have these mutations, the 45% of cells, they must be a subset of the cells that have those 70% mutations, because 70% plus 45% is more than 100%. So they have to be nested within each other. That's called the pigeonhole principle. So we could build this phylogenetic tree of the cancer just by using a single sample. What people make sometimes mistake, they take a biopsy, you all take biopsies, and see a mutation that is clonal, but you need to remember it's clonal in your biopsy. If you take a different biopsy from the tumor, it might not be there, and therefore spatial heterogeneity is very important. Let's move on. Now we could apply the same idea of this phylogic in 2 dimensions. Take sample at time point 1, sample at time point 2, and look at this clustering, before clustering, after clustering, and we could identify clones. This clone changed over time, at 10% at time point 1, and roughly 80% or 75% in time point 2. So this clone is growing. There could be other clones that are shrinking over time. This gives us a lot of information to see where do we look for the drivers, because the drivers should be in this clone of the resistance, not in these clones that are shrinking. So that gives us some clues. And indeed, taking 2 time points is helpful, but it's not helpful enough. The next thing we developed a new version of the algorithm called Phylogic NDT for n-dimensional and timing. And the reason is that our previous algorithm in 2 dimensions could not have been scaled to higher dimensions. It became exponentially more complicated. But a new version of the algorithm can actually work now on many samples from the same patient, and we could start to model the dynamics of clones over time on patients or over space in the body. And there's these clones that are in all the samples, and clones that change over time. This one is growing, this one is growing, this one is shrinking, etc. And this is much more powerful than looking at 2 time points, because things that in 2 time points could have looked the same CCF later on could change their dynamics. One is actually growing and one is shrinking, and you couldn't have distinguished them if you had only 1 sample or 2 samples. Now we need to build a phylogenetic tree. So here's an example with 5 samples in a patient, and we find many of these clusters, which are these clones, but we don't know which ones are the parents of which in the historic development of the cancer. So we developed a Bayesian analysis of these trees, where we try different trees and see which ones fit best the data. And this is all done probabilistic way, and we use the same pigeonhole principle, but now in a probabilistic way. And we're generating an ensemble of trees. There's no one solution that is the true one, because given the data, sometimes we don't know. So we have a distribution of trees that reflects your patient, the history of the patient. So we used this technology recently, together with Kathy Wu, to study CLL even pre-treatment. And what we see, we build this phylogenetic tree, and we see here a clone, this blue clone, is a subset of this orange clone. Interestingly, this orange clone, or whatever pinkish color, has p53 mutation. But this blue clone is a subset of it, had a deletion of 17p, so a deletion of the other copy of p53. And now you can see that the blue clone is growing much faster than the pink clone. So it actually enables, we could measure the first time the fitness of the cells in vivo. We could now estimate what is the growth rate of this clone compared to a different clone. So now we could do that also on patients over treatment if we measure their tumor over time. A third approach to study resistance is to take two different cohorts, not from the same patient. For example, this is TCJ breast cancer. Look at the most mutated genes are PIK3CA, P53, etc. And then compare the frequencies of events to a metastatic breast cancer after kind of resistance to anti-ER treatment. And the most mutated gene is ESR1, which is the estrogen receptor, which is the target of the treatment. And if you look at the pre-treatment samples, it's not in the list. It's very low in the list of being mutated. So that's another indication that ESR1 and actually ARID1A as well are actually drivers of resistance to anti-ER treatment. So now there's a problem. We want to find samples before treatment and after treatment. But typically, you guys don't take biopsies after treatment. Maybe in some clinical trials, after resistance. Maybe some clinical trials do, but most of them don't have the samples after the resistance. So to do that, we develop a blood biopsy approach, where we would take blood samples, and from a 5 mil tube or whatever, 10 mil tube, you could actually get cell-free DNA and perform whole exome sequencing from the blood. And the way we do it, we take the blood, we generate a library of the DNA from that plasma, and then we use ultra-low-pass whole genome sequencing to look for copy number events and predict how much tumor there is in that tube of blood, or in the plasma, in the cell-free DNA, because most of the cell-free DNA is coming from dying normal cells in the blood. And then if it's more than 10%, we could perform whole exome sequencing. So then patients that are metastatic typically have more than, many of them have more than 10%, and then we could do whole exome sequencing. And indeed, when we did it, we show, first of all, that what we find by cell-free DNA is the same in terms of the clonal mutations that we find in the biopsies of the cancer. And we could find here, this is the same example I showed before, these ESR mutations from blood, not just from the biopsy. So with all these tools in mind, we initiated together, we collaborate with IBM Research. We started a project, a 5-year project on studying resistance in cancer in general. And we are actually open to collaboration, and part of the reason that I'm happy to stand here is that I want to invite you all to collaborate on this. And the idea is to have these, to take tumor types and treatments and study and take before and after samples and study the resistance. So we need you. So what are the questions that we're asking? What are the drivers of resistance? Do resistant cells have new vulnerabilities? How do tumors evolve under therapeutic pressure? Can we predict drug resistance mechanisms? Can we find biomarkers of response? Can we predict eventually effective drug combinations? All the data in this project is a fully academic project. All data becomes available in dbGaP to the community, and everyone will be able to access it and do whatever research they want. It has two arms to this project. One is a genomic arm, taking samples either from blood, from frozen, or from FFPE over time on patients, and doing the analysis of exome, RNA-seq, whole genome, single-cell RNA-seq, depending what we need in the study. And there's another arm of it, which is a functional study on cell lines to look for dependencies and vulnerabilities by CRISPR screens or drug screens, etc. And now I want to tell you a bit about the first study that we did with this, we published from this effort. And this is together with Ryan Cochran from MGH that studies GI cancers. And what we did, we took 42 GI cancers that were treated with targeted therapies. And first, we did a biopsy, Ryan did it, the biopsies, and looked for known resistance mechanisms to those drugs. And found that, in many cases, more than half, we didn't find any resistance mechanism. In some, we found one resistance mechanism. In a few cases, we found two. But then, Ryan also collected blood from the same patients. And what we saw that in the cell-free DNA of the blood, we were able to find many resistance mechanisms per patient. In more than a third of them, we could find at least two resistance mechanisms, which we couldn't find by a single biopsy. And when you think about it, it's quite trivial. If you have multiple metastases in the body, there's no reason that the metastases in your liver will develop the same resistance than the metastases in your brain. And there are essentially two different populations of cells, and they could develop different sorts of resistance. So the patient could have multiple resistance mechanisms in their body. And here's an extreme example here, the bottom part there. This is a BRAF a patient that was treated with a BRAF inhibitor. And that patient had 8 different resistance mechanisms. A BRAF amplification, 3 KRAS mutations, different mutations, and NRAS mutations, 2 MAP2K1 and 1 MAP2K2 mutations, all in the MAP kinase pathway seen by blood, by cell-free DNA. When you looked at the tumor biopsy, none of them were detected in the tumor biopsy because they were in different parts of the body. So then we started to look at different parts of the body. And here a patient that got 4 different biopsies. And each one of these biopsies, we could identify different resistance mechanisms, but not all of them. The only sample that could actually find all of the resistance mechanisms was the blood. And we could build these phylogenetic trees, and these, what we call layered pie charts, represent for every sample what fraction of cells belong to which branch in the phylogenetic tree. And as you can see, the pie chart or the pizza at the bottom of the cell-free DNA has many small slices because the cell-free DNA is capturing DNA from multiple parts of the body and therefore samples more lesions. We did the same thing in an autopsy study where we could, this is a gastric cancer that developed FGFR2 fusion treated with an FGFR2 inhibitor and developed multiple different FGFR2 mutations downstream of that FGFR2 fusion. And we could see even people migrating around the world, historically, we could see the migration of cells throughout the body, how based on the mutations that we could measure. And again, the cell-free DNA could actually detect multiple resistance mechanisms not detected in individual biopsies. So to summarize this, we are discovering resistance mechanism, acquired and intrinsic, and biomarkers of them. We could analyze samples of different types, FFPE, frozen cell-free DNA, to perform large-scale studies, which are needed, large-scale studies, and therefore you are here to help. And then ultimate goal is find drug combinations. And then I would say multiple patients host multiple resistance mechanisms. We want to, of course, combine with the functional studies, and that will be done later. Finally, each one of these projects has different steps from initiation to publication and putting in dbGaP. And we are having multiple of these projects, and we invite you to join us to do more of them. And here are our contact information, and we are here today. Brian is here in the crowd. And I want to thank the many people involved in all of these projects, and happy to, question is in the break. Happy to answer questions in the break.